Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I wanted to let you know that we are working on a new program geared towards transitioning from academia to industry. Powered by Dr. GPCR, our new program is called the Life Career Job. The goal is to support you in your efforts to find your next job in industry, plan out your career trajectory, create a life, and support the lifestyle that works for you. If you'd like to be notified when this program becomes available, fill out our short survey on drgpcr.com/career. We are also launching our GPCR consulting services. Soon you'll be able to find the profiles of our carefully selected consultants on our website at drgpcr.com/consulting. You'll be able to find the help you need for your company. Stay tuned. The second edition of the Dr. GPCR Summit will be held between September 13th and 19th, 2021. We're planning a combination of live talks, pre-recorded talks, and live workshops. Visit drgpcr.com/summit for more information. Are you subscribed to our YouTube channel? If not, please subscribe today. It's not only a great way to catch up on our recorded events, such as the Dr. GPCR Virtual Cafe, but it's also a great way to support us here at Dr. GPCR. All right, let's dive into our episode now. Hello, everyone. This is Yamina from Dr. GPCR. I am delighted today to have with me Dr. Randy Hall. He's a professor of pharmacology and chemical biology in the Emory University School of Medicine. Hi, Randy. It's really great to have you here.
Hey, Yamina. It's great to be here. I'm a big fan of the show.
I'm glad that we finally got to do this. And as a first disclosure to the audience, we met by email a couple of months back when we corresponded about a book you've been working on. Can you tell us about the book that you've been working on?
Yeah, so as you know, I was a postdoc in the Lefkowitz lab in the 1990s. And at that time, I was just, I realized early on that, Bob is one of the all-time great raconteurs. He's an inveterate storyteller. And I was, entertained and inspired on a daily basis, while working in the lab when Bob would just hold court and tell stories. And so I thought even at that time that Bob's stories would make for a great book. Back in the 1980s when I was in college, I read a book called Surely You're Joking, Mr. Feynman. It's the memoir of the Nobel Prize-winning physicist Richard Feynman. And I was very inspired by that book, really to consider a career in research. Even though Feynman was in physics and I wanted to go into biology, I still found that book incredibly inspiring. And when I joined Bob's lab, I kind of realized that Bob is like the Feynman of biology, because basically, they're both Nobel Prize-winning giants in their field, and they're both like Jewish guys from New York City, and they're both amazing storytellers. And so I really had thought at that time, and even when I was in Bob's lab, that Bob's stories in his life and all his adventures would make a great book. And sometime shortly after I left the lab in the early 2000s, I was hanging out with Bob at a meeting and had suggested the idea of doing a book that was like the Feynman book. Because Feynman's book was actually co-written with a junior colleague of his named Ralph Layton. Feynman told Ralph Layton all the stories, and Layton wrote them up. And so I proposed to Bob that we could have a sort of a similar model and a similar working relationship where he would tell me his stories and I would help him to write the book. But at that time in the early 2000s, he had like no interest at all in this project, like zero interest. He was so focused on running his laboratory and doing his thing, just was not on his mind at all to do a book like that. And then I guess after he won the Nobel Prize in 2012. We were at a meeting together the next year. I think it's 2013. I suggested again this idea of the book project, and Bob said, "Well, having just won the prize, I have so many invitations and things are just so crazy right now. There's just no way that I could think about it right now." So, but then fast forward to January of 2019. So now this is like a couple of years ago. And I visited Duke that month to catch up on what's going on in the Lefkowitz lab, visit with other friends at Duke, hang out with Bob, very importantly go to a Duke basketball game together because both Bob and I are big Duke basketball fans. And so when we were at dinner before the game, I raised the idea of the book project again. And this time Bob was more receptive. He thought, I'm kind of getting later in my career. And maybe this would be the right time to write a memoir. And we talked about like how it might happen. And I suggested that we could just start talking on the phone for like an hour a week. He could tell me his stories and I could write them up and send them to him to be edited. So we began doing that starting in early 2019. And then we basically wrote the book all through 2019 and then worked on editing it last year after we got a publisher.
Yeah.
That was like during the pandemic, we were taking some time to edit the book and get it in shape. And it was just published in February of 2021. So just a couple months ago, and the reception has been fantastic. So it's been really wonderful to see. Like we've got some great reviews in like Nature and New York Times and other outlets, and then also just random people will write to me and Bob and say how much they love the book and how inspired they feel, and they. So it's been really wonderful to see the reception that the book has received.
It's a great book, and as you know, and the audience knows as well, I sat down in December to chat with Bob about the book, and he's an amazing storyteller. And I was so excited to not only read the book, but I have to disclose this: I did get an autographed copy of the book, which is it's sitting on a shelf with the Terry Kanakin's autographed Primer to Pharmacology book. And I loved reading it. I loved chatting with Bob. And yes, he does have many stories. It's kind of rare. It's not that obvious that people sit down and write books. Can you tell us a little bit about the 1 hour chat that you had with Bob? How did you decide which stories would go into the book, which won't? And how did you go about, once you had that draft, about finding a publisher?
Well, that was definitely the hardest part of the process, was determining, what stories would go in and what stories would stay out, because Bob has so many stories. And each week when I would write up a chapter based on our conversation and send it to Bob, he would immediately reply and say, well, what about this story? Well, what about that story? Like all the things on the cutting room floor, he'd be a little bit agitated that this or that story got dropped. And I had to remind him and say, Bob, if I included every story you told me, this book would be 3,000 pages long. The publisher wants 300 pages, not 3,000 pages. And so a lot of stories had to be dropped. And that triage process was really the only difficult part of writing. But yeah, after we had written a draft, we got an agent, Jim Levine at LGR, and he's a very experienced agent. And he helped us to pitch the book around to several dozen publishers. And a lot of the publishers replied and said, like, oh, we love this book. Like, we think there should be more science books out there. And this guy Lefkowitz has had this amazing life. His stories are incredible. We love the book, but there isn't that big of a market for science books, and certainly science memoirs. There's not that many in the past and the last decade or two that have sold that well. And so there was like a lot of like reticence on the part of the publishers to go in, even though they expressed a lot of enthusiasm for it. But then fortunately, Jessica Case at Pegasus Publishing, she's a real, excellent editor there, and they're kind of a medium-sized publisher. And she was very enthusiastic about the book, like right out of the gate, and just loved it and made an offer on it. And so we just went with her because she was so enthusiastic and seemed like such a highly regarded editor. We were very, very pleased to work with her, and it turned out to be a very good working relationship.
That's great. Yeah, I corresponded very briefly with Jessica as well while we were setting up the podcast with Bob. And I think, yeah, she seems like a really, really good, she's great. She's great. I loved my email exchanges with her. So do you think there is space to write another 300-page book?
Well, Bob has already, given the reception that the book has received, Bob has already been talking about writing a sequel. There were several books by Feynman and Ralph Layton, because after Shirley Joking was a bestseller, then they did several others. They also had stories on the cutting room floor. Mm-hmm. So there were several other books. One that was called "What Do You Care What Other People Think?" And then there was I think even one after that, and there were also like relative bestsellers. And so it is, it is possible, something that we could think about, but that we definitely have material for.
That's why I figured
further books.
When I started chatting with Bob on the podcast, him and I were trying to figure out, okay, how long should the podcast be? How should we go about it? And then I remember that we went way beyond the time that we had agreed on in the beginning, and I felt like there was more to talk about, more stories, and I'm pretty sure that the audience and everyone who's read the book would love to have a sequel and learn more about, hear more about Bob's stories. Mm-hmm. That's fantastic.
Yeah, you definitely know more than anyone how many stories Bob has and how he can just keep talking. When you're talking to Bob, there's never like a pause in the conversation or quiet moments that are like awkward because Bob will just keep talking, and he's such a great conversationalist. Always has something interesting to say, and so you certainly had that experience because it ended up being like a 2-hour plus podcast.
Yes, it did, and I think it's actually now that I know it's the most listened to podcast episode ever that we did here at Dr. GPCR with over at this point almost 600 plays. So at least 600 people listen to it, or multiple people listen to it 600 times, which is fantastic. And it's, yes, you're right. I think Bob has he's naturally outgoing, and he has a lot of stories. What I really loved about our conversation with Bob is that he had great stories to tell, but he was honest. You could tell that Bob is direct. He'll tell you. I loved how he told the stories about the discoveries that he made, the people he worked with, but also how he was honest about his time in the lab versus his time commitments at home. And
yeah, absolutely. I thought he was a great subject to work with on a memoir because he's very emotionally open. Yeah. I thought our sessions were sometimes almost like therapy, where he's just being totally open about so many of his emotions from things that happened in the past. And that's great because I think that's the first and most important secret for writing a memoir is that you have to be honest. That it's not just enough to be interesting or tell the stories that you want people to know. You need to tell, be totally open and tell all the stories from your life and just be honest about things. That to me is what makes a great memoir because it packs some emotional punch. You can tell when someone's being honest in a memoir versus when someone's like holding back and trying to make themselves look good all the time, but none of us are perfect. None of us are, are flawless. And we all have things in our past that maybe, we're not always so proud of. And so, yeah, it's always great that Bob was very open about everything from his past. And I think that's part of what makes it a great book.
I think so too. And that's also what made the podcast episode a really great podcast episode. First of all, it was Bob. Me, when I have to say this, when I first received the email to talk about the podcast opportunity with Bob and look at the book, it's interesting because I started the podcast in the beginning of 2020, and to get to a point where at the end of that same year when I started a project just to see if it would work out, and it made me happy talking to people. Closing up the year with a discussion with Bob Lefkowitz himself, it was just such a great honor. It was a lovely, lovely experience.
Well, I know he enjoyed it, and it certainly turned into a really good and really long podcast. So
yes.
But every minute of it is great. So.
I thought so too. And it's kind of a spoiler alert, but I'm working with you right now and with Bob to get a series of interviews about Bob, and that's all I'm going to say, but we're working on it, and hopefully we'll be able to move forward with that project and get that project out to the audience. I'm pretty sure everyone would love to hear more about who Bob is as a person and how Bob evolved from being the junior professor to the Nobel laureate and way beyond. Super. I'm super excited to see if there will be a sequel. I think there are enough stories to write 2 more books. And how did you come up with the title? That's one thing I wanted to ask.
Yeah, so the title is A Funny Thing Happened on the Way to Stockholm. And that is sort of a play on the Stephen Sondheim play that's called A Funny Thing Happened on the Way to the Forum. That was very popular on Broadway in like the late 50s and early 60s when Bob was growing up. And it was later made into like a very popular movie. And so I think that since that title was something that Bob was familiar with from his youth, then he, after he won the Nobel Prize, he was invited by some student groups and other groups sometimes to give talks about what it's like to win a Nobel Prize, what it's like to go to Stockholm, and all the hoopla and all the grandeur that are associated with that ceremony, and then of course all the sort of the crazy things that happen when winning a Nobel Prize is you achieve like a slightly higher level of celebrity than like a normal scientist, right? Because already even before the prize, of course, he's very well known in the GPCR field and the science world in general. But then after you win a Nobel Prize, you sort of now you begin hanging out at meetings with like Sting and Peter Gabriel and stuff. And it's sort of like a different level of celebrity. And so there's some interesting stories there that he would tell like to the student groups. And so we captured that also in the book, of course. And so that's was the genesis of the title of the book. Was that was also a title he would use for his talks when he would talk to student groups. The title of his talks would be a funny thing happened on the way to Stockholm, where he'd just tell his funny stories about around and after the Nobel Prize.
Yes, before I think he was supposed to give a talk. I was back then a postdoc at Rockefeller University, and he was supposed to give a talk on a Friday afternoon at Rockefeller. On the same week, the Nobel Prize, the Nobel Committee made the announcement, and obviously it got postponed, and we had to wait an entire year for Bob to come back and actually give his talk. And I remember the year after I was still there, and after giving his talk, we went down to the university hangout place of the faculty. I can see it's been a while since I was at Rockefeller, but it's kind of this bar on the campus, and we were sitting around Bob, and there were plenty of students, postdocs, PhD students, and he was telling his stories, and someone from the committee who was with him had to get him out of there because they were going to dinner. But I'm pretty sure that if no one would have come to get him, we would have spent the evening listening to Bob's stories.
Absolutely.
Do you have an idea on what the next book's title might be?
No, I have not discussed it.
I feel like it's going to be a difficult book to come up with a title with, unless once you have the material put back together.
Yeah. I think that's the more natural way, is to write first and try to come up with a title later. Just like we do for scientific papers, right? Usually you do all the work first, and then right when you're about to submit it, you spend a lot of time thinking about the title. So I think it's the same for a lot of books. Agreed
on. So you mentioned that you were a postdoc in Bob's lab. Can you tell us a little bit about your story as a scientist?
Sure. So I guess, I grew up going on a lot of nature walks with my father and watching a lot of nature shows on TV. I was really into all the animal shows. So I was interested from a very young age, in biology. But then I think as I got on and through junior high and high school, I realized that I was more interested in sort of like the mechanisms of how things work rather than just like, watching wildlife per se. And so in college, I began working in a research laboratory. This guy, I was at the University of New Hampshire, and I worked with a guy there named Rick Cody, who we were studying like visual signaling and like photoreceptors, cyclic GMP levels, and like how there's like different compartments of cyclic GMP. Like it's not just that cyclic GMP goes high or low, but there's different compartments of it. It's high in some places, low in others. And so I just found this so fascinating. And so then in grad school, I went to UC Irvine in the neurobiology program there. And I worked with a guy named Gary Lynch, and we studied glutamate receptors, but the ion channel linked receptors, like the AMPA receptor and the NMDA receptor. And so that was my first exposure to working on receptors, and an area that I found fascinating because it's like you're studying that's like the first point of communication between cells. Like that's how cells communicate, right? This one cell releases something, the other cell binds it by a receptor. And so I was so drawn in and captivated by that area that then for my first postdoc, I wanted to kind of stay in that area. And so I went to the Ballin Institute in Portland, Oregon, and I worked with a guy there named Tom Soderling, who was studying the regulation of AMPA receptors and NMDA receptors by phosphorylation. So I began doing a lot of phosphorylation studies there and regulation. But then after a year or two, I realized that maybe my postdoctoral work was a little too close to what I was doing as a grad student, and I think that's a common mistake that a lot of students make is, if you're working in a hot area as a grad student, you think, well, maybe I should kind of stay in this field, right? Because it's like things are hot, and I know the literature of this area. So you do a postdoc that's kind of very closely related, but then that isn't ideal for your development as a scholar, because if you're going to like become a faculty member and show your breadth as a scholar, you kind of need to work on a few different diverse areas, not just one narrow field. And so I decided that what I wanted to do was still work on receptors, because that obviously is a very broad area, but that I should do a second postdoc and work on like a different type of receptor. And so then I chose to work on GPCRs because, of course, this is the biggest family of cell surface receptors. And I wrote to Bob Lefkowitz, and who, of course, was a leader in the field at that time, as he is now. But at that time, his lab had just cloned, the adrenergic receptors and a bunch of other GPCRs. And so, it was definitely a hot lab to go to. Unfortunately, Bob had a spot for me. So I went to the Lefkowitz lab in the 90s. And I think that moment when you, as a scientist, when you change subfields, that often is a very exciting and fertile time for discovery. And my career was certainly an example of that, because having worked on like the NMDA receptors, the big discovery in that subfield at that time was that the C terminus of NMDA receptors, right, has a very specific motif that binds to these scaffold proteins. They're called PDZ scaffolds, like PSD95, that the post-active density protein of 95 kilodaltons. And this is very important for like anchoring of the NMDA receptor at synapses and the way it regulates synapses. And so that was a brand new discovery right at the time that I was moving to Bob's lab. So I was thinking along these lines, and when I joined Bob's lab, I looked at the sequences of the recently cloned beta adrenergic receptors, and saw that basically all of them had these similar motifs that were similar to the NMDA receptor. And I thought, that definitely there could be interesting proteins, scaffold proteins, regulatory proteins, signaling proteins binding to the C termini of these receptors. And so I asked Bob and I asked other folks in the lab, I said, what is known about the importance of the C termini of the, beta adrenergic receptors? And Bob and the others in the lab, they said, that they don't do much. You could, we've done studies, you can remove them and you still get perfectly fine G protein coupling. It's really like, especially the distal regions, they're really not doing too much. In fact, at that time, the Lefkowitz lab was working on trying to get the first crystal structure, right, of a beta of a GPCR. They were working on the beta 2 receptor, and they were just removing the whole C terminus because like who needs it? Just get this out of here. They were focused only on the 7TM region to get the first crystal structure. And so that was how lowly regarded the C terminus was. So they were just throwing it in the trash, like don't even need it to look at the crystal structure. And so I thought, maybe this is actually a ripe area for investigation. If everyone in the field believes that the C terminus of GPCRs doesn't do anything, then I decided to look for proteins that bind to the C terminus of the beta adrenergic receptors and other receptors. And so I took proteomic approaches, yeast-2 hybrid approaches, and was zoomed in on trying to find things that bind to C terminus. And we found a bunch of interesting proteins that bind to the beta 1 subtype, different proteins that bind to the beta 2 subtype, different scaffolds that bind to other GPCRs that were otherwise pretty closely related. And so that was a very exciting, kind of breakthrough in the late 90s, because it led to a lot of insights. For example, there was a lot of literature at that time about differences in functionality between beta 1 and beta 2 receptors. Beta 1 and beta 2 could be expressed together like in cardiomyocytes, yep. Right? And they couple to the same G protein, and they both bind beta arrestins, but yet they have radically different effects on cell physiology, right? So people say, how can this be if these two related subtypes if they couple to the same G protein and both bind arrestins, like how are they having such differential effects on cell physiology? And so, but then if you realize, via their distal C terminus, they bind to other proteins that are very distinct, then that begins to explain how they can have differential effects on cell physiology. And so that was basically my body of work from the Lefkowitz lab was focused on that, trying to study those novel interactions and to elucidate how it explains the differences in subtype functionality between different adrenergic receptors.
And how did the world react to your discovery that actually the C-terminal tail of GPCRs is actually very important?
Yeah, well, there was, I think, a lot of skepticism at first of some of our data. That's normal. I would show the data at Gordon conferences, and you get a lot of pushback from people like the Al Gilmans of the world come by and ask tough questions, and they're a little skeptical about this, But then I think, ultimately our data was convincing. I had a paper published in Nature. I had some other big papers in JBC and PNAS. And ultimately you begin to get acceptance. And then, of course, the ultimate praise is when other labs begin jumping in doing similar studies on not only on the adrenergic, but on all other subfamilies of GPCR. So certainly by the time I became faculty at Emory in the early 2000s, there was so many dozens and dozens of labs that were now studying proteins binding to the C-terminus of GPCRs and showing the functional importance of those interactions. And so just a couple of years earlier, it had been very like avant-garde, but then by the mid 2000s, it was like very like just commonplace, like run of the mill, like so many labs were working in that area.
I love hearing these types of stories because when I started working on GPCRs, this was already known. So to me, thinking that actually there was a period in time where no one knew or no one cared about the C-terminus of GPCRs, and they thought, hey, you can just remove it.
Yeah,
it's just
absolutely.
It's just an interesting realization, knowing that there is actually people who didn't even know what a receptor looked like in the beginning. Mm-hmm. They knew the concept of it, mm-hmm. but there was no real description, and just cloning it was a big advance in the field.
Yeah, and of course, in the Lefkowitz memoir, we go back even further to like the late 60s, early 70s, when people were skeptical that receptors even existed. So that's taking it to a whole other level. To me, that was obviously before my time, and so I really enjoyed hearing those stories from Bob about the pushback that he received, because his idea was, I want to purify receptors and study their properties. And then you had major leaders in the field saying, that's stupid, Bob. It doesn't even make any sense because receptors are just a theoretical construct. They don't really exist as physical entities, but they're just like a pattern of forces in the membrane. A drug binds and to this pattern of forces and causes a change, but there's not a physical thing that's like a receptor. And so obviously we've come a long way in the last 50 or 60 years.
We have, definitely. And then so after your second postdoc in Bob's lab, you ended up as a faculty at Emory University. Mm-hmm. What does your lab work on exactly?
Yeah, so over the years, we've worked on a lot of different subfamilies of GPCRs. So my initial studies were focused on applying the idea of regulation by the C-terminus, to other subfamilies. We worked on metabotropic glutamate receptors, and GABA B receptors, purinergic receptors, a bunch of other subtypes. In more recent years, as I said, that kind of work became, very like standard and run of the mill by say 2010. So now we've kind of moved into studying more a number of orphan receptors, and especially we moved into studying the adhesion subfamily of GPCRs, which I think is a really, really fascinating family.
I think so too. And you had mentioned that you kind of always worked in the realm of the receptor family. Mm-hmm. When was that first time when you were exposed to the concept or to the existence of receptors?
Mm-hmm. Yeah, so the work I did as an undergrad was kind of GPCR adjacent because I was working on cyclic GMP levels and photoreceptors. So that's similar, but not exactly on receptors. But then in grad school, working on the AMPA and NMDA receptors, that's when I first began to fall in love with the idea of studying the initial events of cell signaling. And just find it, it's very simple in some respects because it's like the first event. You're not necessarily looking at all these complicated downstream signaling pathways where you can have so many pathways that have all this crosstalk. So when you're looking at just the initial events that kick off signaling, there's something simple and appealing about that for me. But obviously it's not too simple because now we have concepts like biased agonism, and not every agonist or drug can affect the receptor in the same way. And so to me, it's still a very exciting and vibrant area. And
then throw in their allosteric modulators, and then
absolutely
you've complicated it more mm-hmm. than it was 50 years ago, definitely. So you mentioned, I want to get back a little bit into the same area. So you worked on NMDA receptors, and you were always interested by the concept or the first contact of a receptor with a chemical signal outside the cell. How did you pick up the lab that you first worked in? Did you attend to a class and you got interested in the topic, or how did that happen?
Yeah, I guess so. Before I joined the lab of Gary Lynch, I had read some of his papers because I was going to UC Irvine, and I had read some of his papers. I think I did one of his papers for a journal club when I was in undergrad. And so that's kind of how I became very interested, in the lab's research. In particular, I wasn't interested so much in receptors at that time as I was interested in the Lynch lab in studying synaptic plasticity and the changes that underlie learning and memory. And so that was definitely the genesis of that. But then in the course of that work, then I began to get more interested in receptor biology. But my first, the impetus for joining that lab in grad school was, synaptic plasticity. And I think that's, a lot of students think about, like, when they're going to grad school or when they're going for a postdoc, they think about, like, what lab should I join? Right. It's always the big question.
Yeah.
And I think that always you just got to follow your heart, and whatever you find yourself being interested in, whatever, if you do like a journal club and whatever papers you're picking for your journal club, like that kind of shows you what you're being interested in as a scientist. And so I think you just kind of go with that. For Francis Crick used to call it the gossip test, right? That basically, as a scientist, whatever subfields and little discoveries you find yourself gossiping about, like as you talk to your scientific colleagues, you say, "Hey, did you see this paper in Nature, or did you see this paper in PNAS?" And blah, 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 and you just talk whatever catches your interest, right? Try to realize what's catching your interest, and then go in that direction, because clearly if you're gossiping about it and talking about it, it's something that appeals to you. And so, I guess in my career, I've always tried to adhere to that, to Crick's like gossip test, and just kind of go and find, labs and mentors and research problems that were interesting to me. And that's that was how I got into the Lynch lab in grad school.
It's interesting. You've given many examples on how you went from one stage of your career into the next one, and what comes out to me is that you're always going after something that you're passionate about, and you always picked. It seems to be that you've been picking problems that you were interested in, but also you didn't care what other people were thinking, whether it was done before, it wasn't done, or as you mentioned with the C terminus of GPCRs. What would you tell students that are interested in a certain topic that they're unsure about? Because I feel like back looking at myself 10, 20 years ago, I was a young student, impressionable, and always kind of following what was the high of the interest of that moment. But it took a certain time and maturity and building that confidence to go after topics that I was interested in. Whenever people were telling me, well, that's like with the C terminus, that's
right.
Important.
Yeah, I think that my advice to young scientists is to go for subfields and research questions where there's some mystery. There's some mysteries to be solved. Don't choose subfields where everything seems neat and everything's ironed out and things are very mature. And because I feel like sometimes students are attracted to areas that seem very like orderly and neat. And where things seem like everything's happening as it should be, but it's better to go for subfields where the seas are rough and the sky is dark and there's a lot of mysteries and there's a lot of controversy, like people do not necessarily agree on things. Like those are the subfields where you can make big discoveries, because there's obviously a lot that's not known, but there's a lot of interest. And so I think that's as young scientists are thinking about, labs to choose for grad school, labs to choose for a postdoc. That's good advice, is to go head for the turmoil. Go for subfields that seem like they're in turmoil. Obviously, you don't want to work on things that are too far afield and like where the technology doesn't exist to actually answer the questions that you want to answer. And so that certainly there's a real trick to problem selection, choosing things that are different enough to be interesting, but not so out there that the technology doesn't yet exist to actually address the questions.
Yeah. So basically follow your interests, follow your gut at that point. You'd mentioned that you're currently, you're transitioned to working on adhesion receptors. Are there any specific receptors that you're working on? Can you tell us a little bit more about the projects that are currently?
Sure. I mean, I just think that it's a really interesting subfamily of GPCRs, because there's like 33 members of the subfamily in humans, and they're very exotic receptors. They have huge N-termini extracellularly that can be like thousands of amino acids. All of them have a so-called gain domain. That's like a GPCR autoproteolysis inducing domain where they cleave themselves, but then the two pieces, the N-terminus and the 7-TM region, they remain physically associated together. And of course, the reasons for that have long been obscure and are still much debated in terms of why you'd have these self-cleaving receptors, but that then stay together. And in some of our earliest work, so this is almost a decade ago now, some of our earliest work on these receptors, we were just, because there's a lot of disease genes in the family, right? With the large number of genetic diseases are caused by mutations to adhesion GPCRs. And so we were interested in one of those examples that was GPR56. Shen Piao, who was then at Harvard working with Chris Walsh, she had showed that GPR56 mutations cause an interesting neurological disorder. And so, but of course, at that time, nothing was known about how that receptor functioned, or even if it could couple to G proteins. Right? So, very basic questions like, does this thing even signal? Like, is it actually a GPCR? It looks like a GPCR, but is it actually a GPCR? So, we were addressing like very fundamental questions like that. And some of our earliest studies were expressing GPR56 like in HEC293 cells, and we could see that it had some activity. It was coupling to G12 and G13 to activate Rho. So that was the first evidence that it actually was, a GPCR. But then we noted that since it had some level of constitutive activity, we speculated that maybe there was like a ligand that was present in HEC293 cells, or also maybe in COS cells. We would also doing some work in COS cells. But these cells might express an endogenous ligand that was binding to the large N-terminus of GPR56 and activating it. And so according to that hypothesis, if you remove that large N-terminus, that should create a dead receptor, right? That wouldn't signal. And so we created a truncated version of GPR56 that was missing the N-terminus, missing that was right up to the cleavage point. But then rather than causing that to be a dead receptor, we actually found that massively increased the constitutive signaling by GPR56. And we found identical data for another member of the family, BAI1, that's the brain angiogenesis inhibitor 1. We showed that a protein that receptor is found at synapses, and also showed that it also couples to G12 and G13. And also showed that when you remove the N-terminus, you get a huge increase in signaling, right? And as you well know, because you're an expert in this field, for most GPCRs, if you remove the N-terminus, you do not massively increase signaling.
You shouldn't, at least.
Yeah, that's not normal activity for a GPCR. And so we were very interested in this. And of course, that also is like a general point, right, to point out for students in research that whenever you find the opposite of what you're expecting, that usually that's a very exciting discovery, right? So sometimes people get nervous about that if you make a discovery that's the opposite of what you were hoping for or like looking for. But in this case, we thought it was very interesting because it suggested a mechanism for activating the adhesion GPCR. So we published some papers in 2011, 2013, in some since that time, basically showing that this is quite general for a number of adhesion GPCRs, and of course other labs have jumped into and shown this for other adhesion GPCRs, that when you remove the N-terminus, that you get this large increase in the signaling activity. And so we had proposed, even from the first paper, was in 2011, we had proposed that this could be a general mechanism of activation for the whole family, that basically you get engagement of this large N-terminus by some kind of ligand, and then maybe it gets either conformationally changed or pulled off and removed. And then that's sort of like pulling the pin out of a grenade, and that activates the receptor. And then so that prediction made almost 10 years ago now has largely been borne out over the past decade, because like I say, many labs studying many of the different adhesion GPCRs have shown that invariably when you remove the N-terminus, right, you get a large increase in signaling activity. And then of course there's been further work on the mechanism by which that happens. So like, Ines Liebscher and Torsten Schoenberg in Leipzig showed, and Greg Tall also showed it totally independently in his work in the USA, that when you remove the N-terminus, you unveil a sort of a tethered cryptic agonist in a way that's sort of like the PAR receptors, right? The protease activated receptors that like thrombin activating, cleaving PAR1. In the case of the PARs, right, it's an exogenous protease that does the cleavage to unveil the cryptic agonist. But in the case of the adhesion GPCRs, they have their own intrinsic proteolytic activity, but the idea is the same. That basically you're unveiling a cryptic agonist, and so that's been certainly some exciting advances in the past few years on adhesion GPCRs, and I think they certainly represent also great drug targets, because they're involved in so many important physiological processes. Many of them have very discrete tissue distributions in the body. that suggests that they could be really good drug targets. And they're certainly druggable because, like Greg Tall's lab has done some screening and found small molecules that bind to adhesion GPCRs. I should point out that there was some skepticism that they could be druggable, right? Because that they have such huge extracellular N-termini. And you talk to a lot of the old drug development gurus from back in the day, and they say, oh yeah, these large adhesion proteins, they're terrible drug targets because like some of the integrins and other large adhesive proteins people have tried to drug, but the interaction interfaces of like a lot of adhesion proteins are so large that it's hard to disrupt or modulate with the small molecules, right? And so a lot of drug development gurus are kind of in the mindset that like large adhesion proteins, like they're not good drug targets. But of course, the thing about adhesion GPCRs, they're not just large adhesion proteins; they're also GPCRs.
Exactly.
Right. So, which are like the ultimate, perfect drug target that so many important drugs have been developed against. And so certainly you've seen from the work of Greg Tall and others that he screened and found small molecules that bind to different adhesion GPCRs, both agonists and antagonists, that bind into the 17M region and can regulate their activity. And then even more recently, so just like a couple months ago, the lab of Jinpeng Sung, who's my scientific little brother, right? he was in the Lefkowitz lab in the era just after I was, and now he has his own lab in China. And he published in Nature, right, that it was the first cryo-EM structure of an adhesion GPCR, that was GPR97. And also showed, interestingly, that GPR97 can bind to certain steroid hormones, like the glucocorticoids, that those bind to and agonize GPR97. So, of course, that raises the interesting question, like, well, what about other adhesion GPCRs? Do other adhesion GPCRs also have small molecule ligands, maybe steroid hormone ligands, which is a fascinating area because there's, as you certainly know very well, over the past couple of decades, there's been a huge literature on steroids activating GPCR pathways, right, via like largely unknown mechanisms. There is previously one GPCR, GPR30, right, also known as GPR, which is the G protein coupled receptor activated by estrogen. That was shown to be shown like a decade ago to be activated by estrogen. But other than that, the ability of other steroid hormones to activate G protein pathways has been largely mysterious. But now Jinpeng's discovery says, well, hey, maybe a lot of adhesion GPCRs can be activated by small molecules, and specifically by steroids. And so, because we and others have been mostly in the mindset of like that the ligands for adhesion GPCRs are mostly large adhesive ligands. But Jinpeng's work shows, maybe they do have adhesive ligands, but they also have some small molecule ligands also. And so there's certainly, with N-termini that large, there certainly is room for multiple ligands to bind. And it looks like that may be the case for many of the adhesion GPCRs.
I find it fascinating when these new discoveries are being made, because you always feel like, okay, we know enough. Now we can take it to the next step, and there's always something new that comes up. And I feel like you had mentioned that adhesion GPCRs have very long N-terminis, and they can be very tissue specific when it comes to their expression. And in my mind, those two things together, and the fact that they're specific and they're a little bit cryptic, and removing that N-terminus completely activates the receptor. I think it's just the right clues that show that, yes, it is a really nice way to modulate the receptors in the context of diseases. And hormones, I mean, they do everything. I tell people, I tell non-GPCR fans, GPCRs control everything. No matter what you do, no matter what disease you're talking about, or no matter what event you're talking about, there is always some GPCR or some group of GPCR that is involved in that event.
Absolutely. And I find it so fascinating that GPCRs, of course, they have been throughout history, the last many, many decades, they've been the greatest drug targets. And I certainly thought, like, let's say back in the 90s when I was in Bob's lab, I thought as we get into the 2000s and into the 21st century, that the percentage of drugs out there that were developed against GPCRs versus other targets would decrease, because there's certainly this whole other classes of proteins that could be great drug targets. And you would think as we learn more about these other drug targets and other classes of proteins, that they would have great drugs too, and you'd have a lower and lower fraction. Because I think back in the 90s, it was very common that people would often say, oh yeah, like 30%, 30 to 40% of drugs that are out there on the market target GPCRs, right? And so I always thought that number would decrease over the years, but actually, ironically and amazingly, in recent years, that number is actually increasing. Because you have just in the last few years, right? You have like the orexin receptor antagonists, for treating sleep, and you have the GLP-1 agonists for treating diabetes, and you have so many other great drugs that have been approved in just the last few years that target GPCRs and are doing new and amazing things. And so amazingly, the percentage of drugs that act on GPCRs is actually going up, not going down. So that shows that GPCRs continue to be, just a premier class of drug targets. And there's still so many orphans out there that we know very little about, and that all the adhesion GPCRs are right for drugging and for developing therapeutics against. It's a pretty large subfamily. So I think there's a lot more in the coming decades. I think there's a lot more great drugs to come that will target GPCRs. And we're by no means done in developing great therapeutics against this family of proteins.
I think we're not only not done yet with developing therapeutics against GPCRs. I think we're not done yet understanding the intricacies of the different receptor families of the receptors. The orphan receptors are a class that we think they're GPCRs. They look like GPCRs, but we don't know which ligands, which natural ligands they might bind. And we're at the verge of discovering what type of diseases, what type of physiological events they're involved in. And I think one key to getting to that understanding is moving the technologies and the techniques that we have currently into the next era where we can better measure receptor function and the consequences of receptor activation. And with that, my next question is, what do you think is missing from the field when it comes to technology and tools and initiatives to better characterize adhesion receptors, for example?
Yeah, it's a great question. And I think my hope is that the next great advance will come in the area of in silico screening, right? The idea that as we get more and more crystal structures and more cryo-EM structures, that we'll know more and more about exactly how drugs are binding to GPCRs. And then based on that information that we'll get from dozens or hundreds of different, drug receptor interactions, we can refine our models, our computational models, to predict. So then when you're studying an orphan GPCR that looks like it could be a great drug target, but you don't know any drugs that bind to it, rather than having to do expensive and time-consuming screens like in the real world where you're screening like thousands or millions of compounds, that you can look at the structure and just in silico, computationally predict what drugs are going to bind to that structure. I think that would be very powerful, because it would just save a huge amount of time and effort to be able to make more. Obviously, people are trying to do that now, but many of the predictions are not that accurate because we just lack the knowledge and lack the base of knowledge about GPCR drug interactions. So, but as every month, as more and more papers are published and more structures are out there, I think it's going to get better and better and better. And hopefully in the next decade, we'll have, something by 2030, we'll have just, really accurate prediction algorithms that can really predict what kind of drugs are going to fit in different binding crevices. And I think that there would just be a quantum leap forward and allow us to really accelerate drug discovery, toward not only GPCRs, but really for all drug targets. But that to me is one of the most exciting, current directions that I think it's going to just explode and become, a dominant way of discovering drugs in the next decade or two.
I think it's definitely one exciting novel way of not so novel, looking at receptor and drug interactions using computational biology, and to the next level would be using machine learning and to predict. Not only show that the molecule binds a certain receptor or a certain subtype of receptor or a receptor in a specific conformation, but also being able to take that information and use it in machine learning studies, and then predict what will happen or what might happen in the future with that interaction and how it will influence cellular function. Definitely.
Yeah, so many companies these days that are just starting up are using some kind of AI technology, right?
Yeah.
So, which I think is incredibly exciting because when you look, for example, what's happened in the chess world, obviously these days we have, no human can beat the top computers, right, in chess. But if you have a human working with a machine, right, like the humans and machines working together, then that team can beat any machine or beat any individual human, even like the top grandmaster. So that shows that when humans and machines collaborate, right, that's really, really powerful because they bring different strengths, right, to analyzing any kind of question, whether it be chess or drug discovery. And so I think what's happened in the chess world will be exactly the same in the drug discovery world. That basically, when you have humans working together with machines and AI, that's going to be incredibly powerful for drug discovery, and it's going to result in just like a quantum leap and just a colossal step forward beyond where we are now in terms of the pace of drug discovery and the kind of really novel insights that we'll have. So it's not that machines and AI are not going to replace the humans, but they're going to collaborate with humans. The humans and AI working together are going to be like an unbeatable combination, just like they are right now in the chess world.
I think so too. I think whenever you're looking at machine learning or AI, the input of the data is very important. The quality of the data that you input in the system is very important, and it takes humans to decide what data do you input in the system in order to get your answer. And I think the ability of using machine learning and computational biology to analyze a large amount of data, a large number of results and experiments from different type of systems is crucial. No one human or group of humans can do the work of a computer when it comes to large amounts of data.
Totally agree.
And then actually we kind of answered that question, but I ask this from everyone. Do you think GPCRs are still good drug targets?
Well, they are.
I think so. I think they all are. What do you think the next hot class of GPCRs might be when it comes to new drugs?
I'm going to have to say the adhesions. Got to go with the adhesion family because for the reasons I mentioned, so many of them are human disease genes, right? And so that shows that the impact that they can have, right, on human physiology. And now that we're making inroads and discovering the first small molecules, I think it's definitely a subfamily of GPCRs that you're going to see really take off in terms of the number of drugs being developed.
You mentioned that one of these adhesion receptors, the BIA receptor 1, when it's mutated, it does induce some disease, some type of disease, some brain-related diseases. What other diseases are adhesion receptors involved in?
Yeah, so GPR56, right, mutations cause a neurological disorder as shown by Shan Piao. But my lab has described for BAY2, there's a mutation that also is associated with a neurodegenerative condition. It's kind of rare, but it's still, there are a few examples of it out there. Mutations in VLGR1, which has the great name of the very large G protein coupled receptor. So that's one that's found in the photoreceptors and also in the cochlea in the ears. That causes mutations of VLGR1 cause a kind of deafness that also is associated with seizures and visual problems. And so that certainly is a very important disease gene. And then there are a number of others that have been linked in genetic studies, for example, to ADHD, and to schizophrenia, and to some other conditions of it that are certainly of interest in terms of trying to develop therapeutics. And
these mutations are usually single mutations, or these deletions, or what kind of mutations are?
Yeah, it's both, but there's a lot of them that are SNPs and are single point mutations that are causing pathology.
And can you tell us how these were first discovered? Was there an effort to look for GPCR, or was it just an effort to look for any mutations?
Yeah, for example, with Shan Piao, she and Chris Wallace were just interested in trying to understand this disorder, which is called BFPP, that's bilateral frontoparietal polymicrogyria, which is like a problem of the wiring of the cerebral cortex. And they were just doing the basic genetics on it. They had no idea if it was a GPCR or something else, and it turned out to be an adhesion GPCR. So quite a few of these human diseases linked to the adhesion GPCRs that have been just found in a totally unbiased way, just trying to do the genetics on some interesting disorder, and then finding, it's a GPCR that is causing it.
What type of molecule would we be looking for if you were to modulate some of these mutations in general?
Yeah, it depends. The mutation we found in BAY2 that leads to this neurodegenerative condition, we showed by expressing that mutated receptor in culture cells, showed that it's massively overactive relative to the wild type receptor. So in that case, obviously, you'd want some kind of antagonist or more likely an inverse agonist, right? That could bind to the mutant receptor and tone it down, or maybe like a negative allosteric modulator that would just dampen down the activity of that overactive receptor. And of course, this is hardly a unique idea, right? That overactive GPCRs, cause disease, because that was, there's been so many examples of that over the years that actually that subfield sort of began. This is mentioned briefly in the book, in the Lefkowitz memoir, but back in the early 90s, Susanna Katechia in the Lefkowitz lab was just doing some fun and basic studies on the alpha-1 receptors, right? And showing that, like, I think she was trying to map the region where they're G protein coupled. And so she was making mutations on like the third cytoplasmic loop. And for one particular residue, she found that any residue she changed it to would massively increase, right, the agonist independent signaling of the receptor. And so then this led to the idea that, that maybe the alpha-1 receptor, and maybe many GPCRs, they're tuned to have low activity, right, in the absence of agonists, and tuned to have, and then high activity in the presence of agonists. And so many mutations then throw the receptor out of tune and make it have signaling activity even in the absence of agonists when they're not supposed to be active. And so that was sort of just basic research, sort of an interesting laboratory phenomenon that Susanna and colleagues showed in the early 90s. But then subsequently, all through the 90s, all through the early 2000s, then lab after lab after lab began reporting, that this disease or that disease is caused by a mutation to a GPCR that causes constitutive activity, right? That you have this agonist and independent, uncontrolled activity of a receptor that's causing pathology. And so that turned out that work from Susanna Katechia in the Lefkowitz lab, it had a lot of relevance, right, to human disease, even though that was far from Susanna's mind at that time. She was just doing basic research. Yeah. But then it turned out to be very relevant to a lot of diseases. And certainly our work on BAY2, this member of the adhesion family, is certainly relates back to that concept, and is in that same vein of research that, when you have mutations that cause constitutive overactivity, that's usually not good, and it's going to cause disease in many cases.
When it comes to this particular adhesion receptor, this BAY2, you mentioned that. So you have this single mutation that causes the receptor to become constitutively active. When relative to a deletion of the N-terminus of BAY2, how does that constitutive activity look like? Mm-hmm. Whenever you have the wild type versus mutant versus truncated receptor.
Right. Yeah, the mutation in question for BAY2 is found on the C-terminus. And so that really gets back to that, as you know, the subject near and dear to my heart, why the C-terminus is so important for regulating GPCRs, right? So obviously we've been very interested in understanding what protein-protein interaction is disrupted or enhanced or modulated by these mutations on the BAY2 C-terminus that are causing overactivity and that are linked to disease. And so we've been very interested in trying to characterize, what exactly is changing and how those are functionally changing the receptor. And so it really gets back to the things I was working on even back in the 90s in the Lefkowitz lab in terms of C-terminal regulation of GPCRs.
How does that constitutive activity of that mutated C-terminal receptor compare to the potential constitutive activity of a truncated one?
Yeah, it looks the same as far as we can tell. Wow. I don't know that it biases the receptor in any way, at least not that we've been able to see. Of course, it's probably when you're studying bias, you're a little bit constrained by the assays that you're doing, and maybe we haven't looked at the right pathway. But as far as we can tell, we're not causing bias, but it's just causing an increase in the same pathway that normally is active by the natural mechanism of activation for that receptor.
Mm-hmm. And is it coupled to G1213 as?
So that one is kind of strange. It couples actually best in our hands to G alpha Z, which is a less commonly studied member of the G protein family, but BAY2, at least in hex cells and several other cultured cells that we've studied, it couples very well to GZ, and activate some pathways that are downstream of that.
It's so interesting because whenever you go out of the realm of GI and GS, then everything gets so much more complicated, because for a long time there were no good assays to quantify the activation of the other G protein.
Yeah, exactly. And so that, it is interesting to get away from the classical GS or GQ or GI signaling. But there's certainly a lot of other G proteins out there, and certainly certain GPCRs are tuned to optimally interact with all different members of the family.
I think that's one of the things I love about GPCRs. It's not only the diversity of the ligands, the diversity of the receptor, but also the ability of receptors to couple to different G proteins, depending on the cellular environment and to the tissue specificity. I think it's just amazing. Mm-hmm. It's just fantastic. We've talked a little bit about, your advice to young scientists when it comes to choosing their path, to choosing their question that they want to work on. Do you have any other advice to junior scientists who are in the field or even outside the field who want to make an impact in the science world?
Yeah, I think, I would say work on important problems. I think that there's a lot of important problems out there. There's also a lot of trivial problems and just very detail-oriented things. And in my experience, the trivial problems aren't necessarily any easier to solve than the really important problems, right? There's a lot of trivial problems that are really hard to solve. And you can spend months or years of your career working on something that's fairly trivial. So if you're going to invest the time and effort, right, in a project, some long-term project, at least it should be something that's really important. And so I think sometimes people are in the mindset that the important problems are probably really hard, and so I'll work on something that's a little more peripheral and it's a little easier. But in reality, there's no connection between the importance of a problem and how easy it is to solve. There's a lot of, pretty important questions out there that would be relatively easy to solve if you happen to get on the right path. And conversely, as I said, there's a lot of trivial problems that are really, really hard to solve and can take you years, and the payoff is not very high. And so I think problem selection is very important for young scientists. But definitely don't be afraid to tackle the important problems, because if you're going to invest the time, make it for something that's big and important.
How would you differentiate an important problem from a trivial one?
Yeah, it's a great question, and I just think that you can gauge that both on your own level of interest, the level of interest of other scientists. When you talk about it with scientists at meetings, right, you can get a sense in the field for what are the big questions that people are finding very exciting and interesting and important. And also, just in terms of the disease relevance also, so you want to work on something that it has some potential, right, connection to disease or pathology or drug development. It's always nice if there's a potential. It doesn't have to be a direct, but at least a potential, clinical angle that you might have in mind. I think that helps to steer you in the direction of something, questions that are big and important and will be very interesting to many folks in the field.
And with GPCRs, it's not difficult to make that connection between some clinical
absolutely
phenotype. Definitely. Mm-hmm. We've talked about, working on the importance of the C-terminal tail of GPCRs. Were there at any point in your career, not only while in Bob's lab, where you discovered something or you learned something that you could qualify as an aha moment?
Mm-hmm. Yeah, I think that for example, the work I described earlier on the adhesion GPCRs of when we removed the N-terminus, thinking that would kill the activity of the receptor, and actually found that massively increased the activity. That was for us was certainly a big aha moment. And I think that's very exciting. It should be exciting for any scientist when you do an experiment expecting one result and you get the exact opposite. Like usually that tells you that you're pointed in the right direction and you should further explore that and go that way. Because if you're surprised by the result of your own experiment, then probably other scientists will also be surprised, right? And that always makes for good storytelling, because then when you write the paper, you can say, so we did this experiment expecting X, but we actually got the exact opposite, right? That's like good narrative storytelling, right? Because it's very surprising. So then you want to do further work to figure out why it is that you got the opposite of what you were expecting. Yeah. And so I think that is again, in terms of it relates back to your question about how do you tell important problems. If you're doing experiments and you get the exact opposite of what you expect on some really key experiment, then it tells you that maybe you're honing in on an important question and an important line of research, because if something's really surprising to you, it'll be surprising and very interesting and important to others.
It's a little bit like Googling something, and you're in Google, and then Google finishes your sentence, and then you realize, actually, I'm not the only one looking to answer that particular question.
Absolutely.
And have you, before you if there are any other aha moments before we go to those, have you ever truncated the C-terminal part of adhesion receptors? What happens then?
Yeah, it's a great question. And we certainly have done some studies like that on some of the adhesions that we're studying, for example, to try to map the interactions with arrestins and other regulatory proteins of interest that we know bind. And so it varies from receptor to receptor, but because it depends on what interaction you're perturbing with certain truncations. And there are certainly some, human diseases that have been linked. You asking earlier about the mutations to adhesion GPCRs. At least some of them are premature stop codons, where you lose a chunk of the C-terminus. And so presumably you're losing some very physiologically important interactions that are going on that C-terminus. And so the answer is basically that it varies a lot depending on the subtype, but the C-termini of adhesion GPCRs, just like for other subfamilies, are certainly very important for their regulation. And the main difference with adhesions is that for many of them, they have quite large C-termini. They have huge extracellular N-termini, but also large cytoplasmic C-termini, and a lot of opportunity for regulation there.
Which is most likely needed, considering that considering the importance of the N-terminus region of adhesion receptors. So basically, your intuition in the beginning, when you got to Bob's lab, about let's see what the C-termini do.
That's right.
It's still something that needs to be.
It's been borne out.
And see that was the typical good, important problem to go after. And at the end of the day, it was something that people were not interested in back then. But then you made a point to show that the C-terminus of a GPCR is actually important to regulate its signaling. And then, so we went through the aha moments, and my last question to you is, but if you have any job opportunities, any positions open in your lab, how do you usually advertise, and how can people get in touch with you?
Yeah, absolutely. So if any students out there are interested, just send me an email. I certainly have some openings coming up, because right now I have a postdoc who will be leaving shortly to take a faculty position, and a grad student who's graduating, and an undergrad who just graduated. And so, I have some folks who are either have just left or are leaving in the next month or two. So I certainly have openings. And so students can just drop me an email.
That's great. And your email address, as well as the link to your LinkedIn profile, will be available on our website, so people can read your papers and then, choose the problem they're interested in and reach out to you.
Sounds great.
And with this, Randy, I want to thank you for your time.
Thanks, Amina. It's been a great pleasure.
Thank you so much. And then hopefully we can get back together and talk about Bob's next book that you will be co-authoring. I would love that.
Absolutely.
All right. Thank you so much.
All right.
Bye.
Thanks.
Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, Alexa Duran, and Ines Pinero. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.